Hey guys, good evening and welcome back again to your Unacademy NEET English channel. Hope all of you are doing great. So let me know quickly in the chats if all of you can hear me, if I'm perfectly audible and visible to everyone. Quickly let me know in the chats with the thumbs ups, with the Josh, as this is going to be the first bio class on this particular channel, right? And you know who is going to take this, yeah? So let me know quickly in the chats if all of you can hear me. Quickly guys, quickly, everyone. Wonderful, wonderful. So people, how are the classes going? All going perfect, yeah? So till now, today you are done with your physics, you are done with your organic chemistry class, and now it's going to be the time for your biology. After the biology class, it's going to be my class for the chemistry as well at 8. Perfect. So people are saying voice is crystal clear. That's wonderful, that's great. So people, so let's welcome our biology educator on this particular Unacademy NEET English channel, and one of the amazing educators, I must say. You are absolutely going to love her teaching for sure. Yes, so let's call up Ambika ma'am. She'll be taking over the mic now.
Hi everyone, you all, and how's the energy? How's the energy, guys? Hi. I hope you all are doing very well. Welcome to Unacademy NEET English, and I'm your biology educator Ambika Sharma, and today I am going to teach you the biology, and even I'm going to teach biology to the Wasim sir as well. Okay, so how are you guys? How are you? Let me know in the chat section. All of you, and all of you, just like the video, just subscribe our channel, and do share our channel as well, guys. Just tell me, is everything fine? Just a minute, guys. Just a minute.
Just a second, just a second. We don't want any glitch furthermore, so we are fixing all the...
Happy evening, everyone. Happy evening. So tell me, how are you? Camera glitch is there, but I think I'm perfectly visible and audible, isn't it? Can we ignore that part? Is that okay? Actually, we are trying to fix the... Literally, we are trying to fix the setup here. So that is why maybe you will face some glitch in the video, but that's fine. That's fine. That is not going to affect our teaching, isn't it? Isn't it? So first of all, tell me, everyone, tell me, how are you doing? How are you doing? Tell me. I'm going to start the session. Yeah, you will get the PDF for sure. Anything else? Arey, don't worry. I will teach you the organic chemistry. Or bolo. Anything else? Chaliye. So guys, see, what is the plan? See, you know that we have only 2 weeks for the NEET examination, right? And in this span, we have planned everything. We basically want to cover the high weighted chapters first, right? And today we are going to start the cell biology, the cell chapter, and the cell division as well. Okay, this is the plan basically. So today we will talk about the most important things of this particular chapter, and along with that we are going to practice the questions as well. Okay, so let me know in the chat section. So are you guys excited for the first biology class? Tell me quickly, jaldi, quickly. Are you people excited for the biology class? Yeah, we will take marathon whenever required. Anything else? Okay, chaliye, let's start the class, guys. So you guys can see here the schedule as well. Just wait. See, you guys can see the schedule here for the biology. So I will take session at 5, right? Today it is for the cell biology. Tomorrow we will be discussing the plant kingdom plus animal kingdom, right? So I will provide this PDF to you in the Telegram group as well. Now, without wasting any time, let's start the chapter cell. And we are going to start with the cell division first, okay? See, first of all, tell me, everyone in the chat section, why do a cell divide according to you? Quickly, tell me quickly in the chat section, guys. According to you, why do a cell divide? What is the need for a cell to divide? Right, I'll ask one basic question, then I'll come to the point. We will practice the most important points today. We will discuss that points only, and we will practice the questions. Don't worry about that, because we do not have much time. So whatever is important, I'm going to target that firstly, okay? So quickly tell me in the chat section, guys. Quickly tell me in the chat section. Quickly. Why do a cell divide? For growth, okay. For increasing volume, okay. For living, okay. Repair, valid. See, what happened basically, you know that cell is the basic unit of life, isn't it? Just allow me a minute, please. Now it's fine. I'm perfectly audible, right? Am I perfectly audible? Okay. So you know that it is the cell basically, which is the basic unit of life, right? Life is going to start from the cell. So when we talk about the cell, it needs to divide. Why? Because of two things. There are two things by which, a cell divide. The first thing is nucleocytoplasmic ratio. You can note it down. It is important. The very first thing is nucleocytoplasmic ratio. And the second important thing is surface volume ratio. Right? These two things are there. Like, let me take one example. You know that very small cells are very active, right? I hope you all know it, isn't it? That when a cell, it is of small size, it is very active. Why is it so? Because when a cell is small, its nucleus size is big and comparatively cytoplasm is less, right? Cytoplasm is less. So nucleocytoplasmic ratio is high. The very first thing. Second thing is that surface to volume ratio. Right, surface to volume ratio. So when a cell is having the small size, at that time, the surface area is more in comparison to the volume. And when that cell, it will increase its size, what is going to happen? The nucleus size will remain same and the cytoplasm will increase. And it is going to disturb the surface to volume ratio, right? Here the volume will be more and the surface area will be less. So all the things that a cell needs, it will not be fulfilled. So that is why it is very important for a cell to divide so that it can survive, right? This is the very, very, very basic reason why the division occurs. This is very first point, okay? Now, the next point. Whenever we talk about the division, actually, what is happening today? The screen is having, big size and comparatively my height is not that much. So right now, I'm not able to utilize that space properly, but now we will make tables and all, and I'm going to write down all the points. So you will get the PDF. Right now, just listen to me very carefully. I'll tell you each and every important point and in between from NCERT as well. So there is nothing to worry about. So this session is basically for the revision. So focus on that. Okay? Focus on that. Okay? Chale, let's start. So whenever we talk about the division, whenever we talk about the cell cycle, what do we have? We have two things. The very first thing is interphase and then we have the M phase. As I said, we will discuss the important things only, but basics as well. Okay? So first thing is interphase and then it is the M phase. So initially what happened, scientists, they start observing the things. Initially the instruments were not that good. So whenever they observed that cell stages now, so they find ki interphase is, just like a resting stage. This is what they observed. Okay. This is what they observed. The interphase is just like a resting phase in which you can say that no activity is happening initially, right? So that is why when it comes to the interphase, we even call it as resting phase. Okay, but later on, bache, when they got, good microscope, when they observed it in detail, then they got to know that it is the interphase, which is the most energetic phase. Are you all aligned? This is the point. We used to call interphase as resting phase, but along with that, we know that it is the energetic phase. It is the most energetic phase, isn't it? We even call it as I phase, isn't it? Just type it in the chat section. So initially, because of that, right, scientists used to call it as resting stage, but actually it is not the resting phase. It is the phase when a cell is metabolically very, very, very active because cell is preparing itself for the division. So actually division will occur in M phase and all that preparation for the division will take place in the interphase. That is why it is also known as the preparatory phase. Okay. It is also known as the preparatory phase. This is something very, very, very important. I'll tell you why. Okay, so when it comes to the interphase, what do we have? We have G1 phase, also known as growth 1 phase, right? We have S phase, also known as synthesis phase, and then we have G2 phase, that is the growth 2 phase, right? That is the growth 2 phase. Okay, this point is clear. And when you talk about this M phase, that is the division, right? Division. So here in the M phase, we will be having firstly, the karyokinesis and then cytokinesis. Is that clear? So I'm going to touch all the basic part. I'm going to tell you all the things to the point, but yes, I will make sure that you understand each and everything. Done? From this chapter, you people will definitely understand each and everything. Okay, so just after the class, do practice the question and do let me know in the comment section after the class that yes, ma'am, we are able to solve the questions. Okay, is that fine? Okay. So, interphase, also known as resting, energetic, I phase and the preparatory phase. So, we have these three phases here. Okay. Now, what's next? You people are preparing for the NEET exam. This is my favorite example. Okay, this is literally my favorite example. You people are preparing for the NEET exam maybe from last 2 years or from 3 years, and even some students are preparing for it from last 4 years. Are you aligned? Is it so now? You all are preparing for the NEET exam, I think from 2 years, 3 years, or some people are even preparing for it for the 4 years, isn't it? From the last 4 years, isn't it? So in that phase, if other people, they will observe it, they will be like, yeah, what are they doing? They're doing nothing. They're just studying the things. They are, just reading the books, right? They don't get up properly. They don't get up on time. Something like that, isn't it? Isn't it? But you know that you all are preparing for your NEET examination, and that NEET examination is just for 3 hours, right? That examination is just for 3 hours. But you people are preparing, right, for from last 3 years or 4 years. Same is the case here. Same is the case here. When it comes to the interphase, it is that preparatory phase, right? So it takes a lot of time. So when you talk about a cell, when you talk about its interphase, you can say that a cell spends 95% of its lifespan, right, in the interphase. Or you can even say that more than 95%. More than 95%, guys, imagine. Imagine. And this M phase is just for, very, very less duration. Like if I talk about the humans, in the case of humans, in every 24 hours, a cell will divide once. Okay, so that division phase is just for 1 hour. Or you can say that less than 1 hour when it comes to the humans, right? And what about yeast? Can you tell me in the chat section, guys? What about yeast? Quickly tell me in the chat section. Quick, quick, quick. Tell me in the chat section. Quickly, I want to see that energy in the chat section, guys. All of you, all of you, all of you. Just let me know. What is the timing for the division when it comes to the yeast? It is seriously, it is an important question. Thank you so much, Jagan. Thank you so much, Bache. Thank you so much for all your support. Tell me. I want to see the answer in the chat section. All of you guys, quickly tell me in the chat section. Exactly. It is for the 90 minutes. And what about E. coli? What about E. coli? That is Escherichia coli, which is a bacteria. What about E. coli? Yes. For E. coli, it is for, any idea? It is for 20 minutes, right? It is for 20 minutes. So from this example, from these examples, basically, what can we learn? That when it comes to different species, different organisms, their division time, even it varies, okay? Even it varies. Now, what is the next point that we need to discuss? Just write down. Okay. Now, let's talk about these phases, okay? So, firstly, G1. Let me draw it. G1 phase. Now, this is something very important. All of you, please focus. I will use this particular diagram and I'll tell you all the important points, okay? I'm going to tell you all the important points here. So, all of you just draw it with me. It's important. Okay, as of now, a minute. G1 phase, then comes the S phase, then comes the G2 phase, and then what do we have? We have the M phase. So first of all, let's focus on this particular interphase, okay? I'm going to talk about this interphase first. Is that okay, guys? Is that okay? Is there any lag? Kids, is there any lag? All good? All good, all okay? Sure? Is it manageable? Can you manage that? No lag is there, na? Okay, fine. Okay, so what are we talking? We are talking about the G1 phase. So you just need to remember one thing only. When it comes to the interphase, just remember one thing. Let's see, the first phase is going to be the G1 phase. As I said, it is known as growth 1 phase and gap 1 phase. Okay, so in this phase, the cell will prepare itself for the next phase. This is what you need to keep in your head, literally. Right? This is the key for all the questions. Okay, so when it comes to the G1 phase, what is going to happen in G1 phase? In G1 phase, cell will prepare itself for the S phase. When it comes to the S phase, cell is going to prepare itself for the M phase. So this is how the things work. Okay, this is how the things work. So in the G phase, what is going to happen? You tell me now. A cell needs to divide. Just take one example. Let's say I need to distribute a book. Right? I want to give one book to my students. Okay. Just take one example. I want to give a book to my students. Okay. So I cannot do that. I can, let's say we have Prem and Kavya. So I'm saying ki Prem, you keep the half portion and Kavya, you keep the half portion. No, we cannot do like that. Right? I need to photocopy that. I need to photocopy then after that I will give the one copy to the Prem, other copy I will give it to the Kavya. Isn't it? Isn't it? Same is the case here. So if I need to divide my cell, so this is my cell, you know that. Before starting this now, let's talk about the ploidy. You know that we have two things. One is diploid, another is haploid, right? So when it comes to the humans, 2N means diploid condition. It contains 46 chromosomes, our genetic material, the DNA. When it comes to the haploid condition, we have 23 chromosomes, isn't it? Isn't it? This number varies from species to species. You know it very well. Okay, so we have two divisions. In one division, what is going to happen? See, cell will divide. We will have two daughter cells. So this is what? This is the parent cell. Then here, what will we get? We will get two daughter cells. Isn't it? Isn't it? You can even use the term offsprings here. So in very first division, this division is equational. What is it? It is equational. So whenever we talk about the equational division, that is mitosis. Right? It is mitosis. So in mitosis, the daughter cells will have the same number of chromosomes like the parent cell. If parent cell is having 10 chromosomes, daughter cell will have 10 chromosomes. If parent cell is having 20 chromosomes, daughter cell will have 20 chromosomes. And we have one more type of division. You all know it very well. Quickly answer it in the chat section. Quickly answer it in the chat section. One division is the mitosis. What about another? What is the name of another division? Quickly guys in the chat section. Answer karo, fatafat. Jaldi. Arey, I'll explain it. Don't worry about that. This is just the basic. We'll come to the cell cycle. We will discuss all that phases and then we will move to the mitosis. But as of now, just tell me the name. Just tell me the name. I want to check your previous knowledge as well. Your exam is there in few weeks. I really want to check that. Exactly. That is the meiosis. Okay. So in the meiosis, what are we going to get? We will get 4 haploid cells. So when it comes to the diploid, means 2 sets of chromosomes are there. When it comes to the haploid, 1 set of chromosomes are there. So all that things we need to maintain, we need to take care. That is why we have this setup, right? That is why we have such kind of thing. Okay, that is why we have the interphase. Because in the interphase, we want to make sure that whatever daughter cells form, they should have the equal distribution of the chromosomes. They should get everything properly. Okay, that is why. So now what I just said, when it comes to the G1 phase, right? When it comes to the G1 phase, in this phase, cell will do all the activities. Cell is going to do all the activities which are important for S phase. In S phase, cell will do everything which is important for G2 phase. In the G2 phase, cell is going to prepare itself for the M phase. Got it? Got it? I'll quote one example. In the G1 phase, what is going to happen? Protein synthesis will occur. You know it, isn't it? Protein synthesis increase. It increases, right? Protein synthesis will occur and it increases. Okay, very first thing. And when it comes to the protein, you know that your enzymes are protein, right? Enzymes are protein. All the enzymes that we need for the cell activity, all the enzymes that we need for the division, right, for the cell, for the DNA replication, that all that enzymes will form here in the G1 phase, the very first point. Along with the proteins, we even need the RNA. So obviously RNA synthesis will also occur, right? RNA synthesis will also increase. And meanwhile, what will be the result? The cytoplasm. The growth of cytoplasm will be there, means the cell will grow. That is why in the NCERT, it is clearly mentioned that cell is going to grow. Whenever it is the interphase, the cell is going to grow. Done? Done? And what is the next part here? In the S phase, you even need ATP. You even need energy. You need enzymes, right? So ATP synthesis will also occur in particular phase, that is the G1 phase. So what do you have to remember? What is going to happen in G1 phase? The protein synthesis will occur, the RNA synthesis will occur, the ATP formation will be there, and cell is going to increase its size as well, right, bache? And the another point that you need to mention here is that this phase is the most variable. Just write it down. It is the most variable phase. Its duration can change. Right, it depends upon the demand. Okay, its duration can change. Now come to the S phase, which is also known as the synthesis phase. Guys, you have seen many questions in the paper from this particular part, isn't it? From the S phase, I think you all have seen many questions in the paper. Now, as I said, in the G1, cell is preparing itself for the S phase, and in the S phase, it will prepare for the G2 phase. So the very important thing that will occur in this S phase is the DNA synthesis. What is going to happen here, bache? The DNA synthesis. You can also call it as DNA replication. So what is going to happen in this phase, bache? The 2C DNA content will become 4C. The 2C DNA content will become 4C, but this is very important. Number of chromosome remains same. Okay, but number of chromosome will remain same. Just write down the point right now. Then I'll tell you what you have to do. What you need to take care here in this particular part. Right? Just write it down. So what is going to happen? DNA synthesis. 2C DNA will become 4C, and then number of chromosome will remain same. So this is the most important thing. It is an important question. It is a MCQ. Write down. It is the MCQ. I'll explain why is it happening. But write down. Okay. Along with that, what will happen? Centriole duplication. Centriole duplication will also take place here. But just put a star mark here, bache, and note down. This is the case of animal cell only, because centriole is present only in the case of animal cell. That's all. Okay. Just do it. Done? Is it clear? Is it done? Any doubt? Guys, you're not able to hear me properly. Is it so? Is it fine? No, right now we are not talking about the equational division or the reductional division. Right now we are just talking about the interphase in general. Okay, so G1 mein all that activities will occur, and in the S phase, DNA synthesis, DNA duplication, but number of chromosome will remain same. The second important point is centriole duplication, but centriole is only present in the animal cell, right? It is only present in the animal cell. So that is why you have to put a note here. Centriole duplication will only take place in the animal cell, and in which phase it will occur? In which phase it will occur? In the S phase only. Okay? In the S phase only. Is that clear? Done? Sure? Okay. Now, after this S phase, G2 phase, then comes the M phase. So just write it down, bache. Even the histones. You know na histones? You know na DNA is very long. In the case of humans, it is about 2.2 meter long. So we need to coil it. We need to coil it, right? It will coil over the histone proteins. So that histone proteins will also synthesize in this S phase. Just note down, right? Histone synthesis will also occur in the S phase. Chalo, now I'll ask one question. Just take one example. You people are having a question in the NEET examination, and that question is, when will the centriole duplication take place in the animal, in the plant cell? What should be your answer? In which phase of cell cycle the centriole duplication will occur in a plant cell? What are you going to answer? Quickly tell me. What are you going to answer? Quickly, quickly tell me. Might be you will get such brainstorming questions as well. Tell me. Tell me. Very good, Nitesh. Excellent. Very good, Pooja. Noise, Sharat. Very good. Guys, I am saying that in plant cell in which phase centriole duplication will occur, you should say, ma'am, no. In the plant cell, there is no centriole. So your question is wrong. Right? So you need to tell me, ma'am, your question is wrong. Okay. So just take care of such things in the NEET examination. You have to read the question carefully. You really need to read that question carefully. Okay? Done? So now, let's talk about the G2 phase. Now we know that in this phase, right, after this phase, we will be having the M phase where actual cell division will occur. So in the G2 phase, what will happen, bache? Again, the RNA synthesis will be there. Again, the protein synthesis will be there. And we know that after it, cell will divide. So all that organelles duplication, the content for organelle duplication will also occur. Okay. What will happen here? Here in the G2 phase also, bache, what is going to happen? The organelle duplication will also occur. So up to this point, all clear. And moreover, there is one more thing. After G2 phase, there will be the division. Spindle formation will also occur. So for spindle formation, we need that microtubules. So microtubule formation will also occur in G2 phase. So it is an important point. So whatever is important, I'm going to put a star over there. Okay. Whatever is important, na, I'm going to put a star over there. Okay. Done? Yeah, even I'm taking class after so long. Okay. Done? Sure. So up to this point, all clear. So now there is no need to cram. You just need to remember whatever is required for S phase, that will be synthesized in G1 phase. Whatever is required in G2 phase, like DNA and all, DNA duplication, that will be synthesized in S phase. Whatever is necessary for the division in the G2 phase, all the things will occur. Like RNA synthesis, protein synthesis, and in the M phase, in the division, we need to divide the cell. So microtubule formation will also occur here. Okay, the microtubule formation will also occur here. And yes, one more thing, bache, ATP synthesis. ATP synthesis. Done? And then finally come the M phase where first of all, your nucleus will divide. That is known as karyokinesis. Okay. And then the cytoplasm will divide. That is known as cytokinesis. Clear? Clear? So up to this part, all clear? If there is any doubt, just let me know in the chat section. Just let me know in the chat section. Quickly, everyone. Quickly. Explain, Prem, how the spindle formation will be there in plant cell. But right now, tell me. All that okay? Yes, even ATP is going to synthesize here. Definitely. ATP is going to synthesize here for sure. Okay? Done. Now, one more thing. Let's take one example. See, what is my main agenda basically? This is a chapter in which many students, they face problems. Right? They don't understand that how DNA is getting doubled and the number of chromosomes is same. They don't even know what is the actual meaning of equational and reductional division, right? Because you cram the things, you don't understand it. Okay, you don't understand it, you just cram it. Ki, okay, in the NCERT it is written, so we have to follow it accordingly. No. In my sessions, I will tell you the logic behind whatever is very, very, very important. Right now, we are going to put emphasis on that only. Right? And later on, after that, whenever we will start, our batches for the next year, for the NEET 2024, then one by one in detail, I'm going to explain everything. Okay, just keep it in your head. Now, I am going to clear that why is it so that DNA is getting doubled, but the number of chromosomes is same. So, I'm going to quote one example. Let's say this is a cell having 4 chromosomes. Okay, it is a cell having 4 chromosomes. Let me draw it. Okay, done. It is a cell and this cell is having how many chromosomes? This cell is having the 4 chromosomes, isn't it? 4 chromosome. So what is going to happen? Actually, in the interphase, everything, the DNA is not condensed. The DNA is not coiled over the histone proteins. It is placed just like the threads, right? It is placed just like the threads. It is not at all coiled. But right now, just to quote one example, I'm telling you, just imagine in a cell we are having the 4 chromosomes and they all are condensed. Okay, the first point clear? Seriously, is there any issue in voice, guys? Actually, serious. Tell me in the chat section. Am I not perfectly audible and visible? Huh? Just let me check. Okay, voice is perfect. Okay, okay. So, I'm quoting one example that we have a cell having the 4 chromosome, 1, 2, 3 and 4. Now, what is going to happen, bache? After the interphase, especially after the S phase, this is the scenario. Okay, this is the scenario. This is going to happen after S phase, yes. It is not a chromatid, it is a chromosome. And here, it is also a chromosome, the chromosome in which DNA is doubled. Right, in which DNA is doubled. So can I say that after S phase, the number of chromosome is same, but when it comes to the DNA, it is doubled. 2C will become 4C. Can I say that? Can I say that? Yes. I can say so, isn't it? Isn't it? So first doubt clear? First doubt clear? So definitely you will get one question from this particular part. 110% you people are going to get question from this particular part. So kindly note it down. Note it down. For sure you will get question from this particular part. Okay. So this is all about the interphase. This is all about the interphase. Now particularly we will be discussing the M phase. So if there is any doubt, do let me know. If there is any doubt, do let me know. I'll be teaching about the G0 phase. I remember. Tell me, any doubt? Sure? Can we go ahead? Can we go ahead? Duration. See, G1, in G1, the duration is variable. It depends upon the need. Okay, that is what you need to remember. The exact duration, the minutes, no, you don't need that. You don't need that for NEET. Okay, done? So here you can see G1, S and G2. Now we will be talking about this mitosis. But before that, we have one more phase, phase is there, and that is known as G0 phase. Right? Uh-oh. See, here we have the M phase, kids. After M phase, there will be the division, then G1 phase. But we have one more phase also, that is known as G0 phase. Okay, this G0 phase is also known as cell arrest phase. What is it? It is known as cell arrest phase. We even call it as quiescent stage. What is it? It is the quiescent stage. Cell arrest phase and the quiescent stage. Actually, what happened? In our body, not every cell is required to divide, right? We have the examples as well, like we have the heart cell. You all know about the heart cells. We know about the neurons, the nerve cells, the longest cell of the human body. Are you guys aware of it? Are you people aware of it? Yes or no? Yes or no? You know about it, na? Ki the nerve cells, the heart cells, they do not divide. Okay, so what is going to happen in such cells after M phase? For initially, they will enter in the G1 phase. This is a very important point that you people need to remember. That cells will enter in the, after division, all the cells will enter in the G1 phase. But if they will not get signal to divide, then what is going to happen? They will leave G1 and then they will enter in the G0 phase. Firstly, they will go in G1 phase, they will grow their size, the cytoplasmic growth will be there, and after that what is going to happen? If they are not getting the go-ahead signal, then what will they do? They will move to the quiescent stage, that is your G0 phase, right? That is your G0 phase. But being in the G0 phase, but if they are in the G0 phase, that doesn't mean they will not be active. Write down this point. Cell is metabolically active here. Just write it down. Cell is metabolically active here. If a cell is in the G0 phase, it will not divide, but still it is in the active stage. Metabolism is too high because metabolism is very important for a cell to survive. Done, bache? Done? So any doubt here? Any doubt here? Okay. So this is what you need to remember. So it is the another MCQ. As I said, we are going to target the most important things today. So this is the another MCQ. Just write down. You will surely get questions. You will surely get questions from this particular part. 110%. Okay? Done? Chaliye. So now let's start the M phase. Just start the M phase. Done, bache? Karyokinesis and the cytokinesis. So what is the meaning of karyokinesis? Karyo means nuclear. Kinesis means division. Okay, karyo means nuclear. Kinesis means division. So what is going to happen in the karyokinesis? There will be the nuclear division. What will happen in the karyokinesis? There will be the nuclear division. So here in this particular phase, we will be having PMAT. What is going to happen here? PMAT. Write down everyone. What will happen here? PMAT. So what do you understand by PMAT? P stands for prophase. M stands for metaphase. A stands for anaphase. And finally, we will be having the T. T stands for telophase. Is that clear? So write down the trick in the chat section quickly, all of you. Quickly write down the trick in the chat section. Quickly. So what do we have here? We have the prophase, the metaphase, the anaphase, and the telophase. So this is the PMAT. This is the PMAT in the karyokinesis. Okay? Clear? Clear? And finally, the cytoplasmic division will be there. And this cytoplasmic division is different in plant and animal cell. We will discuss it. But first of all, let's focus on this PMAT. Okay? So, bacha, it is very, very, very simple. Trust me. This portion is literally very simple. Oh. Guys, am I visible? Kids, am I visible? Guys, am I audible? Just allow me a minute. Okay. Now, hold on. Okay. Fine? Sure? So I think now I'm perfectly audible and visible, isn't it? Isn't it? Okay. So can we start now once again? See, actually, what is happening? We are, putting all the things together. We are trying to manage everything here. So that is why you are facing glitch in, our sessions. So don't worry about that. By tomorrow, everything will be fine. Everything will be fine for sure. Don't worry about that. Okay, don't worry about that. Okay, so what were we talking? We were talking about the karyokinesis, and in the karyokinesis, we were talking about what? We were talking about the PMAT, right? Prophase, metaphase, anaphase, and telophase. Guys, whenever a cell divides, okay, as I said, in the interphase, initially, the DNA is just like that. Okay, the DNA is just like that in the form of threads. It needs to get condensed, the very first point. The nuclear membrane needs to get, dissolved. Okay, the chromosomes need to form. They need to come to a proper plane so that we can divide it equally. It's very simple. No need to cram anything here. Just imagine one thing. Close your eyes. You are thinking of a cell. You are thinking of cell division. So what is going to happen? Initially, the cell, the cytoplasm, the DNA was, lying down, right? It was just laying down. So we need to coil it first, we need to condense it first, we need to put it on that equatorial plate, and then we need to divide, that's all. So what is going to happen in prophase? Tell me in the chat section. Right now we are just talking about the mitosis. We are not talking about the meiosis here. We are just talking about the mitosis. So what is going to happen in the prophase here? Just tell me quickly. Quickly. Very good, Prem. The condensation of the chromatin material will be there. The first point is condensation. What is going to happen, bache? The condensation of chromatin material will be there. Done. So obviously, nuclear membrane, it will disappear, isn't it? Isn't it? All the cell organelles, they will disappear, isn't it? Yes or no? Isn't it? Isn't it? Tell me, isn't it? Now, along with that, what is going to happen? The centrioles, if it is an animal cell, then centrioles start moving to opposite poles. So any doubt here? Do you have any doubt here? Quickly tell me in the chat section, all of you. Any doubt? Sure? So this is the diagram. See, slowly, slowly, the nuclear membrane, it will disintegrate. The condensation is going to start. And as I said, the centrioles, they will start moving to the opposite pole. Is that clear? Is that clear? Done? So just remember one more thing. This is the DNA. Now it has to become like this because already S phase is over. Okay, already the S phase is over. Done? Done? So here we just need to remember two words. Just write down. The first word is anestral. The second word is amphiestral. The first word is anestral. The second word is amphiestral. Do you know the meaning of these two words? Yes? Do you know the meaning of these two words? Ma'am, what is the difference in early prophase and the late prophase? But initially, just imagine, initially what is going to happen? The nuclear membrane disintegration will just start. The centrioles will just start moving to the opposite poles and the condensation will just start. But when you talk about the late prophase, all the things, they will be done, bache. Okay, they will be done. Done? Okay? So, anestral. Now, as I said, there is no centriole in the plant cell. So, when it comes to the plant cell mitosis, it is going to be anestral, right? It is going to be anestral. And when you talk about the mitosis in the case of animal cell, it is going to be amphiestral because here we have the centriole. Now, how the centriole and the ester word is related, let me tell you. See, the centriole is made up of microtubules, okay? The centriole, it is made up of microtubules and that microtubules are in return made up of tubulin protein, right? Centriole, it is made up of microtubules and microtubules are made up of the tubulin protein. So when these centriole, they will move to the opposite pole. What will they do? Some fibers they are going to radiate. Okay. Some fibers they are going to radiate. And these fibers, they will give the appearance like, like a star. That is why we use the word aster. That is why we use the word aster. So what is going to happen? What is going to happen in the late prophase? These asters will radiate. Along with that, spindle fibers will also form. But in the late prophase, your chromosomes, they will not get arranged in that equatorial plate. They are still scattered. Okay, they are still scattered here and there. They are not aligned properly. They are still scattered here and there. The first point clear? Tell me quickly in the chat section. This point clear? Yes. Now I'll explain how the spindle fiber will form in plant cell. But as of now, tell me. All the points clear? Quickly. Tell me. Guys, tell me quickly in the chat section. All of you. All of you, quickly. We are going to practice the questions as well. So tell me. I want to see the response in the chat section with the thumbs up. Yes. Done? So amphiestral is the word for the plant, for the animal mitosis, and estral is the word for the plant mitosis. So plants, they do not have the centrioles. Animals, yes, they do have the centrioles. And that centriole is going to radiate these fibers, right? So that is why we are going to call it as amphiestral mitosis. Aster formation is there. Aster formation is there. This is a very important point. Okay. Now, the next point. Just say, I think 2 or 3 students were asking, ma'am, how is it possible that in the plant cell, they do not have the centriole, but still the spindle formation is there? So let me tell you, when it comes to the centriole, actually, this is not a centriole. This is a structure like this, where you are having 2 centrioles covered with the pericentriolar material. What is going to happen here, bache? There will be 2 centrioles which are placed 90 degrees to each other. They are covered with the pericentriolar material. And together we call it as this particular structure. We call this structure as centrosome. What we used to call it? We used to call it centrosome or cell center. Done? Centrosome or cell center. So mainly for the formation of that spindle fibers, right? Mainly for the formation of that spindle fiber, this pericentriolar material is required. Actually, this pericentriolar material helps in the formation of that spindle fibers. Okay. So because of the presence of centrioles in animal cell, these spindle fibers are properly arranged, right? They are properly placed. But in the case of plant cell, although they do not have the centriole, but still they have that pericentriolar material, and that pericentriolar material will again do the polymerization of tubulin. They will form microtubule, they will form the spindle fiber, but that spindle fiber is not aligned properly. Okay? That spindle fiber is not aligned properly. That is the point. Done? That is the point. So this point clear? Quickly. This point clear? Okay? Done. So now what is going to happen in the metaphase? I'll explain with the diagrams, and the diagrams are important. You are going to get questions from the diagrams only. Now, metaphase is something very important, and bache, you know what? It is only the prophase that will be different in mitosis and meiosis. Trust me. If you understand prophase, rest all the phases, whether it is metaphase or it is anaphase or it is telophase, all the phases are very easy. It is the prophase that you need to understand. And when it comes to the cell division, it is the prophase which is the longest phase. Okay, it is the prophase which is the longest phase. Just remember it. So now what is going to happen in the metaphase? All your chromosomes, now chromosome is just like that. They are aligned. They will be aligned on this. It will be aligned on this imaginary plate. We'll call it equatorial plate. We will call it metaphasic plate. Okay. All the chromosomes, whatever chromosomes are present in the cell, they will align themselves on this equation. Okay. They will arrange themselves in the center of a cell. Okay. So that is why we call it as equatorial plate or the metaphasic plate. This is very important. And now here, the very important question that will come in your paper is, see, how that spindle fiber will attach to this chromosome. Just look at it very carefully. This is the chromosome. We know that in the center here we have the centromere, isn't it? Here we have the centromere. So this particular centromere, it is covered with two disc-shaped protein, something like this. Something like this. See, this is the centromere, na? So this centromere is covered like this. This centromere is having these disc-shaped proteins, and we used to call it as kinetochores. The word is kinetochore. Sometimes in the paper, they will write kinetosomes, right? They can write something else also. It is basically the kinetochore. What is it? It is the kinetochore. So what is going to happen here in the metaphase? The spindle fiber, they will attach to this kinetochore. They never get attached directly to the centromere. This is something very important. As I said, another MCQ. So, bache, I am going to target all the important questions today. That is why I am majorly focusing on the points from where, question is coming from the last 10 years. Okay, because we have less time now and we have to complete the syllabus, but we have to complete it in a crisp way. That is why I'm highlighting these points. Okay, so just note down. So, it is the kinetochore to which your spindle fibers will get attached. Always. Always means always. Now, this is the metaphase. So it is the phase in which you can, check the shape of the chromosomes. It is the phase in which we can count the number of chromosomes as well. So in the NCERT, there is a line that shape, the number of chromosome, in which phase we can check it in the metaphase. So is that clear? All of you. Is that clear? Done? Tell me, is that clear? Clear? Sure? Okay, so this is the metaphase. Now, one more point. Do you know how much energy will it take to move one chromosome to the opposite poles? Can you tell me in the chat section, all of you? Do you have any idea of it? Because after metaphase, now we will be moving to the anaphase. Just remember the trick. Prophase, metaphase, anaphase, telophase. Prophase, metaphase, anaphase, telophase. So after metaphase, we need to move to the anaphase. So in the anaphase, we are going to split it. So I'm asking for the energy. Do you have any idea that how much energy it will take to move this chromatid or chromosome basically to the opposite poles? Yes? Guys? Quickly. All of you answer the question. I'm waiting for your response. So, what happened first? The condensation started. The organelles disappeared. In the next phase, all the chromosomes, these condensed chromosomes, they are going to arrange here. Okay, they will be arranged here in the center. Okay, now next phase is what? Next phase is the anaphase. Next phase is what? It is the anaphase. What is it? It is the anaphase. Now in the anaphase, just look at it. What is going to happen, kids? Here, this particular chromosome, it will split. So can I say that in the anaphase of the mitosis, the centromere will split, the chromatids will separate? Tell me, can I say so? Yes or no? Just follow the sequence. Can I say that when it comes to the anaphase, it is the centromere which will split? It is the chromatid that will move to the opposite poles? Yes or no? Tell me in the chat section. Quickly, tell me in the chat section. And we are talking about the mitosis here. Yes. These are the points that you need to note down. Yes or no? Yes or no? Definitely. This is about the anaphase. So, here the centromere will split. The chromatid, they will start moving to the opposite pole. Okay, they will start moving to the opposite pole. And you people are right. It takes around 30 ATP. We need 30 ATP to move one chromosome to the opposite pole. So just imagine if in a human cell we have the 46 chromosomes. So you can keep it 46 into 30. That much energy we need for the division. And that is the reason why in the, human reproduction wala topic, when it is the ovulation, at the time of ovulation, the secondary oocyte, it is in that stage, in the metaphase to arrest. Now you can relate it, right? You can relate it. Why that arrest is there in the secondary oocyte? Why that arrest is there in the metaphase 2 stage? Because it takes a lot of energy to move these chromosomes, these chromatid to opposite pole. Just imagine 30 ATP for one chromosome, right? For one chromosome. Done? So this is about the anaphase. Okay. So one more thing is there in the anaphase, bache. You know that when it comes to the chromosome, different shapes are there. If their arms are equal, then this is the centromere, which is also known as the primary construction. So if these arms are equal, then it is going to be metacentric chromosome, right? If the centromere is towards, one end, a little away from the center, submetacentric, right? If it is towards the end, telocentric, you know about it, isn't it? So when it comes to the movement of these chromatids, right? Just remember it. I'll draw it. Just remember, this is going to happen in the anaphase. So actually the splitting will occur. So just look at it. Sometimes you will find V-shaped structure. Sometimes you people are going to find L-shaped structure. So it depends upon the shape of the chromosome. If that chromosome is having the equal arm, so just imagine like someone is holding me from my waist and that person is pulling me. Just imagine. So if I'll keep my arms like this, so it is going to make the V shape. That is why we are having different shapes. Sometimes V shape, sometimes L shape is there. Okay. And even sometimes T shape is there. Okay. That is why we are having the different shapes. So now from anaphase, we have telophase. So telophase is just opposite to that. It is the simplest phase ever. So it is just opposite to prophase. So can you tell me what type of changes will occur in the telophase, guys? Can you tell me what type of changes are going to take place in the telophase? It is just opposite to the prophase. So what will happen? These chromatids, now they are not chromatids, now they are the chromosomes. They will again decondense. Again the nuclear envelope will form. Again the cell organelles will form. Right, bache? So that type of things are going to occur here. Yeah, V shape is there, L shape is there, sometimes J shape is there. You're right. Okay, done? So any doubt? Any doubt? Tell me quickly. Do you have any doubt here? Exactly, your nucleolus will reform, the Golgi apparatus will reform, all that organelles will appear once again. So that is why we used to say that when it comes to the telophase, it is just opposite to the prophase. What is it? It is just opposite to the prophase. Clear? Clear? Okay. So I'll send the PDF. Don't worry about that. So now the next is cytokinesis. Okay. Next is cytokinesis. I already told you about the karyokinesis. So I will explain it with the help of diagrams, and you can see the diagrams here. You all can see the diagrams here. It is very clear. You can quickly revise this topic. So see the condensation, all that arrangement, the movement, and finally this thing. Okay. Finally this thing. Done? Fine. So all the diagrams and points you will get. I'll share the PDF in our Telegram group. You all can join the Telegram group. Okay. So now we will be talking about the cytokinesis. So after karyokinesis, there will be the cytoplasmic division. See, if that division will not occur, just imagine this is a cell, nucleus is dividing, okay? And cytokinesis is not occurring, just nucleus is dividing. We will be having so many nuclei in a cell, isn't it? We will be having so many nuclei in the cell. So then our cell will become multinucleated. Right, our cell will become multinucleated. So this condition is known as cynocytic condition. What is it? It is the cynocytic condition. What is it? It is the cynocytic condition where cell is having so many nuclei. We also use one more word for it, and that is syncytium. Again, it is a PYQ, that is a previous year question. So we have the cynocytic condition, multinucleated condition, and we have the syncytium. Okay, syncytium. Yes, you are right. When you talk about the coconut water, in the coconut water, what do we have? We have free nuclei, right? That is free nuclear endosperm. Okay, done? So now let's talk about the differences in the cytokinesis in the animal cell and in the plant cell. We know it very well when it comes to the plant cell. Plant cell is having the cell wall, isn't it? Plant cell is having what? Plant cell is having the cell wall. So obviously because of that, the cell division, the cytoplasmic division, it varies. Okay, it varies. I'll quote one example. When it comes to animal cell, when it comes to the cytokinesis, a cleavage furrow will form. What will form? A cleavage furrow will form. And what is going to happen here? Actually, actin filaments will head. Just write down this point. When you talk about the cytokinesis in animal cells, what is going to happen? The very first point is the formation of cleavage furrow. Write down, bache, you will get question from this part in the paper for sure. Right? Formation of cleavage furrow will be there. What will be there? Formation of cleavage furrow. So how that furrow will form? It will start from periphery. From periphery, then it will start moving towards the center. From periphery, from boundary, it will start moving towards the center. And for that, which protein is helping? It is the actin. Which cytoskeletal element is helping? It is the actin. So it is the another MCQ. You can write it down. When it comes to the animal cell, cleavage furrow will form. It will form from periphery towards the center, and for that we use the word centripetal. Right? So centripetally, the cleavage furrow will form and grow. Okay? Centripetally, the cleavage furrow will form and grow, and with the help of actin protein. Is that clear? These are the questions that are going to come in the NEET exam. You don't have any need to, check any other question. That's all. Now, when it comes to the plant cell, so we know that in the plant cell we have the cell wall. So now what will happen here? So in the plant cell, there is a difference actually. In the animal cell, in the telophase, by the end of the telophase, the spindle fiber, all that, it will disassemble. Okay, you can write it down this point. So what is going to happen in the animal cell? In the animal cell, right? In the animal cell, in the late telophase, the spindle fibers, they all will disassemble. They will disintegrate. Your that centriole is at the opposite pole. You can write it down, okay? But when it comes to the plant cell, even after the telophase, your spindle fiber will remain like that. Okay? Your spindle fiber will remain like that. So what is going to happen? Just imagine, this is the spindle fiber. Some Golgi vesicles, they will come here in the center. They will fuse. Now, collectively, this structure, because here we have the nucleus. Here we have the nucleus. Here Golgi vesicles, they are fused. So these Golgi vesicles, they are going to form the structure here, and that structure is known as cell plate. What is it? It is the cell plate. Right? What is it? It is the cell plate. And together this structure, this spindle fiber, this cell plate, together we call it as phlegmoplast. What is it? It is phlegmoplast. What is it? It is phlegmoplast. So here you can see the development of the cell plate is from the center towards the periphery. From the center towards the periphery. So what will be the word here? It is centrifugally formed. Okay. Done? This is the key word here. You need to note it down. Okay. You need to note it down. Okay. So it will, from center, it will move towards the periphery. And after that, obviously you know that the cell plate, in future, it is forming the middle lamella, which is enriched with calcium and the magnesium pectate. Okay? It is enriched with calcium and the magnesium pectate. It is another MCQ. Right? It is another MCQ. So when it comes to the mitosis, you are not going to get any question beyond this part. You can even practice the question and you guys can check. Okay? So revision done. Revision done. If there is any doubt from the mitosis, let me know. So now you got it. Why do we call mitosis as the equational division? Because parent cell will, if parent cells are having 20 chromosomes, daughter cells will also have the same chromosomes. So why do we call mitosis as the equational division? Because daughter cells are having same number of chromosomes like the parent cell. Daughter cell is having same number of chromosomes like the parent cell. Done? Done? Any doubt? So if it is all clear, then I want to see the thumbs up in the chat section. Quickly, quickly, quickly. I want to see the thumbs up in the chat section. And do like, do share the video as well. And be quick, guys. Be quick. Done? Okay. So here you can see the difference as well, the cleavage furrow and all. Okay, fine. So now we will be starting with the meiosis. Okay, but before that, just answer this question. Quickly answer this question. Which one of the following statement is incorrect for the interphase stage? Just answer it to me. Just do it quickly, guys. Do it quickly. Which one of the following statement is incorrect for the interphase stage? What should be the answer here? And you are going to find such easy questions in the paper. Quickly. Obviously, it's very easy. The answer is going to be C, right? So, in the interphase, which statement is incorrect? So, bache, while attempting the NEET paper, please read the question carefully. Please read these statements carefully, right, bache? Because if we ignore such parts, now obviously we are going to mark the incorrect answer. So, what is happening here? Here they are asking the incorrect statement. So, yes, it is a period of great metabolic activity, right? Obviously, the metabolism is very high because cell is preparing itself for the division, just like your NEET preparation. While preparing for the NEET, you people put a lot of efforts because you want to make sure that there should be no error, right? There should be no error in your NEET exam. You want to score good marks in the NEET exam, isn't it? Isn't it? So that is why you prepare well, okay? So, second statement, also called a preparatory phase. Yes, I told you. I told you that is why I was telling you it is the I phase, resting phase, preparatory phase, even the energetic phase, even the invisible phase. Next, it covers over 95% of the total duration of cell cycle. Yes. Done? Sure? Okay. Chali, now the next phase. Okay. Post-mitotic gap phase. Try to answer this question. Post-mitotic gap phase is characterized by all except, we are talking about the post-mitotic gap phase. Post-mitotic gap phase. But in the next lecture, we'll be talking about the plant kingdom and the animal kingdom. All the important things from plant kingdom and animal kingdom. Right, because we have, very limited number of days. So whatever chapter we are going to learn, we will focus on the most important topics. Yes, tell me post-mitotic gap phase. Here I'll relate one more thing from the NCERT. I'll relate one more thing here from the NCERT. See, in the NCERT it is written that I phase is the Anji. Do you remember that? In the NCERT, it is clearly mentioned now when we talk about the interphase, we keep it in between two M phases. Isn't it? We keep it in between two M phases. Yes or no? Do you remember that line? So if I mention the I phase here, in the I phase, we are having G1, we are having S, we are having G2, right? In the NCERT, it is mentioned now that interphase is present in between the two successive M phase. Do you remember this? Interphase is present in between two successive M phase. So, in the interphase, we have G1, we have S, we have G2. So, can I say that G1, it is coming after M phase. So, can I say that G1 is the post-mitotic gap phase? Yes or no? Post-mitotic means after mitosis. Can I say that it is after mitosis? So, can we use the word post-mitotic? Of course, we can. And when it comes to the G2, because it is before M phase here, so I am calling it as pre-mitotic gap phase. Okay, pre-mitotic gap phase. Done? Done? So, from here you can learn two important lines from the NCERT. The very first is that I phase is present in between two successive M phase. The second important point is that G1 is the post-mitotic phase, G2 is the pre-mitotic phase. If you know these terms, you guys can answer this question. And here the answer here is synthesis of histone protein. Okay, it will not take place in the G1 phase and it is again a PYQ. Okay, again a PYQ. Chaliye, next question. Okay, very easy. Which of the following is not a diploid cell? I think you all know it, isn't it? Which of the following is not a diploid cell? Yeah, we'll do plant physiology, we'll do genetics, right? Guys, I have already provided you the schedule. Even I will share it once again with you. So don't worry about that. As of now, just focus here and answer the question. All of you, just answer the question. And literally, you should share this link with your friends as well. Connect more people. Connect more people. Quickly. Exactly. Which one of the following is not a diploid cell? Zygote. Definitely diploid. Microspore mother cell. Whenever the word is mother cell, it is diploid. It is the cell in which meiosis will occur. Okay. Primary oocyte, 110% diploid. And when it comes to the ovum, it is the gamete. So it is the haploid cell. Okay. It is the haploid cell. So now can we revise the meiosis very quickly? Anna, we should now? Tell me, we should now? We should revise the meiosis, isn't it? Should we start? Should we start? Yeah, bache, we'll provide you the mock test, we'll provide you the test series, we'll provide you everything. But as of now, just focus here. Just focus here. We told you now that we all are here with a team and we will make sure that you get selected in your dream medical college. We want the NEET 2023 selection of our students as well. Fine. But for that, you need to be sincere. You just need to follow these sessions. You need to be regular. If you people will be regular now, I'll make sure, right? Even we will make sure that you get maximum marks in your paper because we will share the tips, the tricks, the strategies as well, right? The strategies as well. And that is why I chose this particular topic because after this, we will be targeting the genetics as well. And for genetics, you need to understand the mitosis, and that is why that important points are important. Important points I'm highlighting, and I will share the PDF in the Telegram group as well. Okay? Okay? Okay, I'll cover the cell too. Don't worry. So now we are going to talk about the meiosis. As I said, what is meiosis? It is the reductional division. What is the meaning of reductional division? The meaning of reductional division is the one which is going to reduce something. And here, what will it reduce? It will reduce the chromosome number. Okay, what will it reduce? It will reduce the chromosome number. And why this division is important? Because we need it for the formation of the gametes. And gametes are haploid in nature, isn't it? Gametes are haploid in nature. For this particular division, this is what you need to remember. You have a diploid cell. In that diploid cell, first of all, meiosis 1 will occur. You will get 2 haploid cells. And then comes the meiosis 2. After that, you will get 4 haploid cells. Okay. Now, what's the point here? So, by looking at it, can I say that here in the meiosis, there are 2 rounds of nuclear division? Important points I'm talking about right now. You already know about the interphase. Now, we are going to discuss the important points here. And once again here, the prophase 1 is very important. If you understand prophase 1, then everything is easy for you. Okay? Okay? Done? So, here what do we have? We have one diploid cell. So, when it comes to the mitosis, mitosis can even take place in the diploid cell. Even it can take place in the haploid cell. Just write down this point. When it comes to the mitosis, it can even occur in the diploid cell. It can even occur in the haploid cell because some lower plants, they are haploid. Their main plant body is haploid. So, in their case, mitosis will occur. But when it comes to the meiosis, it will only take place in the diploid cell, right? It will only take place in the diploid cell. So, here you can see we are having a diploid cell. So, first of all, meiosis 1 will occur, right? Meiosis 1. So, you can write down this point. When it comes to the meiosis, there will be 2 rounds of, this is all NCRT bache. 2 rounds of nuclear division and cytoplasm division. Just write down. 2 rounds of nuclear division and 2 rounds of cytoplasmic division. But DNA replication will take place once. Okay. But DNA replication will take place once. Is that clear? Yes or no? It's a PYQ. Is that clear? So here you can see the first round where the karyokinesis and cytokinesis will occur. Then comes the second round where the karyokinesis and cytokinesis will occur. But the S phase, the interphase will take place before that. Okay? 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 So the DNA will again be 2C will become 4C. And why do we write here the 2C and 4C? It is just, simply signifying that DNA will get doubled. Just imagine 2C means 100 grams. So double means 200 grams. 4C means 200 grams. That's all. Okay? That's all. So up to this part, all clear? Yes, all of you, tell me in the chat section. All of you, quickly tell me in the chat section. Quickly. Done? Okay. So these two things, these 2 points are very, very, very important. So how are we starting? We are starting with the meiosis 1. The basic thing is same. PMAT. Here in the meiosis 1, it will be prophase 1, metaphase 1, anaphase 1, telophase 1. What will happen in the meiosis 1? Prophase 1, metaphase 1, anaphase 1, telophase 1. So the trick is same. That is PMAT. Right? But here it will be P1, M1, A1 and T1. Okay? The sequence will be same. Now, after this, there will be meiosis 2. Again, prophase, metaphase 2, anaphase 2 and then telophase 2. Okay? Then telophase 2. That's all. Fine? That's all. So let me write it down. Interphase. Actually, this flowchart is important. All of you kindly draw it. Then the meiosis 1. For meiosis 1, I'm using this M1. Then comes the prophase 1, metaphase, anaphase, telophase. When it comes to this prophase 1, here we are having the substages. So just remember this thing, bache. Just remember this thing. This prophase 1 is the longest, right? It is the longest phase. When it comes to the meiosis 1 or meiosis 2, it is the longest phase. And overall in the division, it is the longest phase. Okay? It is the longest phase. Okay? Done? So when it comes to the prophase 1, we have the trick here. Little Zara, please do not dance. What is the trick here? Little Zara, please do not dance. All of you write it in the chat section quickly, or simply you can see LZPDD. I'm writing it here. LZPDD. LZPDD. All of you repeat it. Little Zara, please do not dance. Because sequence is important. Again, you can get a question from this part. Little Zara, please do not dance. Little Zara, please do not dance. All of you just write it in the chat section. So what is it? It is L stands for leptotene. Z stands for zygotene. Pachytene. Diplotene. And then comes the diakinesis. Then comes the diakinesis. So what do we have? Leptotene, zygotene, pachytene, diplotene, diakinesis. So this is the sequence. This is the sequence. So this is important. So after this, M1, anaphase 1, telophase 1. Done? Done. So after this telophase now, kids, what we used to have, after this telophase now, there comes one more phase, a kind of gap phase. We used to call it interkinesis. Okay? Then after that, once again, prophase 2, metaphase 2, anaphase 2 and telophase 2. What am I saying? I'm saying that when it comes to the M phase meiosis 1, after prophase 1, then metaphase 1, anaphase 1, telophase 1, after this telophase 1, there will be a gap period. We used to call it interkinesis. Means the inter means in between, the gap between two divisions. What is it? It is the gap between two divisions, right? So after this interkinesis, there will be prophase 2, metaphase 2, anaphase 2, telophase 2. So here in this phase, basically that haploid cells, they will again divide. They will form 2, 2, 2, 2 daughter haploid cells. So it is just like mitosis. Okay, this stage, this is just like mitosis. Right? So here what do we have in between? Interkinesis. Just remember, in the interkinesis, there is no DNA replication. Nothing at all will happen. right? Nothing at all will happen. So this flow chart is literally important. Kindly note it down. Okay. So because of, my height, I cannot reach here properly like Wasim sir and, that can write properly. So right now, please manage. Please manage. Okay. Chaliye. So let's start with the prophase one. So yes, another important point. Another important point. Guys, here, you will definitely get question from this particular part. For sure, you will get MCQ from this part. For sure, you people are going to get question. For sure. In the NCERT, it is given na, that in these sub-stages, what exactly is happening basically. So yes, question will definitely come from that part, but I'll give you the hint here. See, you remember one thing. From the mitosis, you have learnt one thing, that when it comes to the prophase, in the prophase, ultimately chromosome condensation will take place. What is going to happen? Chromosome condensation will take place. And second important point that you need to remember, in the meiosis, what do we want from meiosis? We want the haploid cell. We want to reduce the chromosome number to half. So just relate these two things. Whatever you already know, you just need to relate it. You just need to, you just need to frame it in a sequence. That is the thing only. Right? That is the thing only. So what is happening in the prophase? Ultimately, we know that there will be the condensation. Nuclear membrane will disintegrate. The spindle fibers will form, means the centriole will move to the opposite pole, definitely. But along with that, here in this particular prophase 1, in these sub-stages, two very important things will take place. Two very important things will take place. The very first thing is pairing of the homologous chromosome. Here you will learn these words. Pairing of homologous chromosomes. That is synapses. That is synapses. Second most important thing that is going to happen here in this prophase 1. It is the crossing over. It is the recombination. Are you getting my point? These 2 points are literally very, very, very important, my dear kids. Okay, these 2 points are literally very, very, very important. Are you clear? Are you clear? I'll tell you the sequence. So you can mention here that when it comes to the prophase 1, longest phase it is. Okay, what is it? It is the longest phase. What is it? It is the longest phase. So write it down in the chat section. Write it down in the chat section. So very first stage, you know it very well. You know the sequence. It is the leptotene. Along with leptotene, you can also call it as leptonema. Leptonema. Okay, leptonema. So chromosome condensation will start here, and this stage is also known as bouquet stage. Are you aware of it? This stage is also known as bouquet stage. I'll explain why. But tell me, do you know about it? Tell me, do you know about it? Then I'll explain why is it called as bouquet stage. Okay, fine. So, in the leptotene, again, nuclear membrane disassembly is starting. Basically, what is happening here now? From one end, all the chromosomes are attached like this. Especially in the case of insects, we observe such things. Okay. In the case of insects, we observe such things. What is happening here? All the chromosome, their ends, they will join together to the one side of this nuclear membrane. Okay? They will join together at the one side of this nuclear membrane. They are going to form the, see, here you are having some attachment plate. So it looks like a bouquet. And you observe such things in the case of insects mostly. So that is why the first stage, the leptotene, is also known as the bouquet stage. Is that clear? That is why the first stage, that is leptotene, it is also known as the bouquet stage. The first point clear? The first point clear? Now, next, we are having the leptotene. Next will be zygotene. And it is LZPDD. It is also known as zygonema. So here in this stage, the very important point is synapses. What is going to happen here in this substage? The very, very, very important things, the synapses will occur. What is going to happen here? Synapses, also known as syndesis. What is it? The pairing of homologous chromosome. What is going to happen here? What is going to happen here, Ruthika, Anika? Yes, the pairing of homologous chromosome. So the very first question, here is ki ma'am, what exactly is the homologous chromosome? I'm not assuming that you know it. I'm going to tell you this. So, this is important. So, kindly listen to me. So, what is happening basically? When you talk about the humans, you know that we have 46 chromosomes, isn't it? All of you know that we are having the 46 chromosome. So, here in these 46 chromosomes, 23 chromosomes are from mother, 23 chromosomes are from father. Because we all are the result of the fertilization, na? There will be a sperm, an ovum, it will fuse, the zygote will be there. From that zygote, we all will grow because that is the totipotent cell. So 23 chromosomes are there in sperm, 23 chromosomes are there in ovum. They are going to fertilize. They will form the diploid zygote, isn't it? Isn't it? And we all are the product of that zygote. Right? We all are the product of that zygote. So what is happening here when it comes to the zygotine? The synapses will occur. So synapses is the word for this pairing of homologous chromosomes. So when we use the word homologous chromosome, just write down. Homo means same. What is the meaning of homo here? Homo means same. Like our mother has contributed the chromosome, our father has contributed the chromosome. So the chromosomes having same centromere position, right? Having same centromere position, having same shape, plus they are having the same genes. Listen to me very carefully. Each and every word is important. You guys will get question from this particular part. Okay, you people are going to get question from this particular part. So what are the homologous chromosome? Homo means same. The chromosome having same centromere position, right? Having same shape, having same genes. Genes are same, but alleles can vary. Genes are same. Furkana bache, genes are same, but alleles can vary. Means if here we have the gene for height in the father's chromosome. So again in the mother's chromosome also, you will find the same gene here, but allele can vary. Maybe it is for the, here we have the capital T, here maybe we are having the small t. So anything can happen. So that is why I'm saying genes are same, but alleles can vary. So when they will get paired, this is known as synapses. Okay, this is known as synapses. So why? Why will they get paired? What is the reason? Obviously, if I want to join two things together, I need to put a glue in between. I need to put the Fevicol in between, isn't it? Isn't it? I need to do that. Okay. So here, when these two homologous chromosomes, they will come closer. So in between, right? Or you can imagine it in this way. Let's say, this is the mother's chromosome. This is the father's chromosome, right? They are paired. How will they get paired? Because in between, they will be having the complex. That complex is known as, bache, synaptonemal complex. Which complex is there? Synaptonemal complex. Synaptonemal complex. Which complex will be there? Synaptonemal complex. So this complex is having the RNA, the proteins, even see these lateral elements, DNA is there. Okay, so this is going to cause that zipping. This is going to attach these chromosomes together. So this is important. The question will come from this part. Very good, Himanshi Thakur. He has mentioned the correct answer in the chat box. Ki, ma'am, it is the ribonucleoprotein complex. What is it? It is the ribonucleoprotein complex. So are you getting it, guys? Quickly type it in the chat section. All of you. Just quickly tell it to me in the chat section. Quickly. It is the synaptonemal complex. Ribonucleoprotein complex it is. Done? Sure? Sure? Okay, fine. Now, synapses is done. Okay. So here you need to understand one more thing, kids, and that is when you talk about this particular chromosome, both the arms, because they are attached to same centromere. Let me change the color. See, both the arms, because they are attached to the same, centromere. What we used to call it? We used to call it sister chromatids. What we used to call it? Sister chromatids, because both the arms are attached to the same centromere. Now come to this pink wala arms. They are also the They are also attached to the same centromere. So they are also known as, they both are the sister chromatids. But when you talk about this pink and yellow, this one, they both are not sister chromatids. They are non-sister chromatids, right? They are non-sister chromatids. So if you have any doubt from this part, do let me know, because again, the question will come from this part. And you can mark my words, just check whenever you will get your NEET 2023 ka paper, now you will find out. Okay, ma'am was saying ki this question will come from this part. This question will come from this part. Definitely you will get these questions in the paper. I'm so much confident about it. Right? I'm literally confident about it, guys. Okay. Okay. And see what Unacademy NEET English is saying, ki hit the like button, yaar. Hit the like button quickly. You will get everything. We'll practice the questions here. Don't worry. So what is this? These are the non-sister chromatids. Okay. So in the zygotine, your synaptonemal complex will form, which will result in the pairing of the homologous chromosome. Okay, which is going to result in the pairing of the homologous chromosome. Now the next important stage is the packetine. Next important stage is what? It is the packetine. It is the stage when the recombination will occur, when the segments will get exchanged, right? And this is the reason why sexual reproduction is better. We used to say na ki for the variations, mutation and recombination is important. I think you all are well aware of it, isn't it? Isn't it? You all are well aware of it, that when you talk about the sexual reproduction, we always say that sexual reproduction is better than asexual reproduction. Sexual reproduction is advanced than asexual reproduction. Why is it so? Because from the sexual reproduction, there will be the variations. The result of sexual reproduction is variation. Why? The meiosis, the packetine, the recombination. The crossing over is responsible for the variation. And that crossing over will take place here in packetine. So packetine, it is the longest sub-stage of the prophase 1. Again a PYQ. It is the longest sub-stage of prophase 1. Just write it down. Longest sub-stage of the prophase 1, which? Packetine. Okay. Packetine. So packetine mein what will happen? The crossing over will take place. What is this crossing over? It is the recombination. It is the recombination. So with the help of recombinase enzyme, this will occur. So what do you need to remember here? Again, see this. This thing will occur. Now you must be thinking, ma'am, what is that? This is basically the exchange of segments. Right? They say, I am not ditto like my father. I have characters from my father as well, from my mother as well, right? I am, in me, I have, some characters from my mother. I have some characters from my father. Why? Because of this recombination. So what is going to happen in this recombination? There will be the exchange of segments, right? There will be the exchange of segment. And how that exchange of segment is possible? It is possible with the help of enzyme recombinase, right? It is possible with the help of enzyme recombinase. It is possible with the help of enzyme. All of you just write it in the chat section. I want to see the energy, guys. I want to see the energy. Be ready for these type of sessions. We all are going to start from 11 a.m. in the morning and you will get your last session till, I think, 11 p.m. easily. So in that we have to cover the syllabus. We have to revise as well. We need to practice the papers as well. We need to practice the questions as well. So yes, in these last 13 to 14 days, you need to practice a lot. You really need to practice a lot. If you are feeling drained, you can take a rest for 10 to 15 minutes. But again, you have to study because we are running out of time and we have to compensate that. We have to study. So are you guys with me? Right? Are you that much motivated? Because we all are motivated as a team, as teachers, that we are going to give our best to them. Are you people ready? Tell me quickly in the chat section. I want to see that energy in the chat section. Okay, quickly. All that, fire and all the thumbs up, I want to see it in the chat section. Okay, so here you can see what is going to happen, the exchange of segment. How? With the help of the enzyme recombinase. So actually before this recombination, right, just let's say at this point the exchange of segment will occur. So before this recombination, at this point, a structure called recombination nodule will form. What is going to happen here, bache? A structure called recombination nodule will form, and that will be the point at which, you know, these non-sister chromatids of homologous pair of chromosomes will exchange their. Okay, these non-sister chromatids of homologous pair of chromosome will exchange their segments. So this is the key word here. Always remember, it's a PYQ as well. When it comes to the crossing over, it is the exchange of segments. But it's a PYQ. All of you just write it down. It is the exchange of segments in the non-sister chromatids. In the non-sister chromatids of homologous pair of chromosome. Done? In the non-sister, right? So exchange of segments in the non-sister chromatids of the homologous pair of chromosomes. And before that, what is going to happen here? The recombination nodule will form. Okay? So what will be the result of crossing over? You can see it here. I hope all your doubts are clear. See, this yellow one will get some pink part. This pink will get some yellow wala part. So actually the exchange of segment is there. That is responsible for the variation. That is responsible for the variation. Just note down. So guys, after this recombination, it doesn't mean that these pair will get separated. No, it will remain like that. It will remain like that. And you know the point at which the crossing over has done, especially at that point, they will remain connected, right? They will remain connected. You will see some X-shaped structures. What type of structure you people will see? You all will see some X-shaped structures. And we used to call it as, can you write it in the chat section? What is it? Chiasmata. What is it? Chiasmata. That's X-shaped structure. The point which is showing, this is the site from where the crossing over has been done. Right? That X-shaped structure. It is chiasmata. What is it? All of you just write it in the chat section. All of you quickly write it in the chat section. Right? It is the X-shaped structure that is known as chiasmata. That is known as chiasmata. So this is going to happen in the packaging, the recombination, the crossing over, the exchange of segments. Fine? The exchange of segments. So after packaging, what do we have? Which one is the next substage? Which one is the next substage? See this packaging, the crossing over will occur. Now the next is diplotin and then comes the dikinesis. So what is going to happen in the diplotin? What is going to happen in the diplotin? Yes? Quickly write it in the chat section. What will happen in the diplotin? Obviously, there is the diplotin. Dissolution of synaptonemal complex, yes or no? This is given in NCERT. And again a repeated question. There will be the dissolution of the synaptonemal complex, isn't it? Isn't it? There will be the dissolution of the synaptonemal complex. So what is going to happen here, bache? The synaptonemal complex will start dissolving. But that doesn't mean that this pair will get separated. No, it is going to, it will not happen here. Actually, the sites, you know, where the crossing over has been done, at that point, that chromosomes will remain attached. Okay, that X-shaped structure will form. That chiasmata formation will be there. But yes, the dissolution of synaptonemal complex will definitely be there in the diplotin. Right? It will definitely be there in the diplotin. Just mark this point. Mark this point. And trust me, the questions are going to come from this particular part. Okay? So, synapses ends, bilkul. Chiasma is visible within the bivalent. I'll tell you this term. Bivalent. Okay? Okay? Now, after this diplotin, there will be the dikinesis. Now, what is happening? Just say, let's say, to draw, you know, a lot of diagrams. And I really like that as well. Just say, let's say, it is the point at which the crossing over is going to take place. Just imagine. It is the point at which the crossing over is going to take place. Just keep it in your head, all of you. Huh? This is the point at which the crossing over is going to take place. So obviously, what we learned, we have learned now, here at this point, after the exchange of segments, there will be the formation of X-shaped structure, that is chiasmata. Do you remember this? Do you remember this, guys? All of you. Do you remember it? Yes or no? Of course you know that. So now in the last stage, that is your diakinesis. That is your diakinesis. It is LZPDD, na? Leptotene, zygotene, pachytene, diplotin, diakinesis. So now in the last substage, there will be the terminalization of chiasmata. Terminalization of chiasmata. So let's say your chiasmata is formed at this particular point. It needs to move to the ends. That is the meaning of terminalization of chiasmata. Okay, it needs to move to the end. Okay, it needs to move to the ends. Just initially it was here, now it will move to this part. We cannot say that chromosomes are totally separated, no, because this pair need to arrange itself on that metaphasic plate, right? On that metaphasic plate. So that is why it is important. Done? That is why it is important. So up to this part all clear, guys? Up to this part all clear? Just tell me quickly. Up to this part all clear? So these are the most important things from this particular chapter. Now just one last topic and this topic will be over then. Next, after this diakinesis, what will happen? There will be the metaphase 1. So now we need to focus there. So now you people are going to get one homework, and that is you need to check the images from the NCERT. Okay. You people need to check the images from the NCERT. That is very, very, very important. In the NCERT, there is one, image na, where it is mentioned transition to metaphase. That is very important. That you have to check. Fine? Fine? Okay. So now, the comparison. It is basically a comparison slide and it will clear all your doubts. Write down all of you. Just write down. Meiosis 1. This comparison slide will clear everything. Everything. Done. Now, when it comes to the metaphase 1, this is the thing, bache. Let's say a cell is having 6 chromosomes. Let's say we are taking the example of a cell having 6 chromosomes. All of you please answer in the chat section. It's a repeated question. Let's say a cell is having the 6 chromosomes. So in the metaphase, right, if it is the mitosis, this is how the chromosomes are going to arrange themselves? Yes or no? This is how the chromosomes are going to arrange themselves? Yes or no? Guys, quickly tell me yes or no. It will happen. It will happen. Now, what is the difference in the meiosis 1? What is going to happen in the meiosis 1? Why after meiosis 1 we are having the haploid set? Right? The daughter cells are haploid there. There is a reduction. How is it possible? Because when it is the metaphase 1, kids, then at that time, there will be the pair. Right? You people will see a pair here. That is the beauty of this meiosis. You people are going to see the pair here. See. That will be the scenario. This is the thing that you need to note down. Just imagine in the final exam, in the NEET 2023 paper, you people are having a question. There is one diagram, there is another diagram. And in the question, they are asking, out of these 2 diagrams, which one is the metaphase 1 of meiosis 1? Right? Which one is the metaphase 1 of meiosis 1? So you need to understand it. So look at this. When it is the simple metaphase of mitosis, right? Correct. When it is the simple metaphase of mitosis, just look at it very carefully, kids. All the 6 chromosomes are arranged like this. There is no pairing. There is no pairing. And even in the anaphase 1, the centromere will split, the chromatids will separate. they will move to the opposite pole. But when it is the metaphase 1, because we need to reduce the chromosome number to half, so there will be the 2 metaphasic plates, there will be the arrangement of pair here. Okay, okay, so this pair is known as, see, bivalent, because it is a pair, so we call it as bivalent. And even because there are 4 arms here, we also use the word tetrad. Okay, we also use the word tetrad. Are you getting my point? We also use the word tetrad. Now, you use your brain and just tell me one thing. Do you think after metaphase 1, there will be the anaphase 1? Yes or no? After metaphase 1, there will be the anaphase 1. Yeah, yes or no? Quickly, quickly, quickly. Quickly. Quickly, guys, quickly. After metaphase 1, there will be the anaphase 1. Yes or no? Yes or no? Definitely there will be. So now just look at this image. So tell me, in the anaphase 1, will there be the splitting of centromere? Will be the splitting of centromere? Will there be the splitting of centromere? Just tell me quickly, all of you. Quickly. Quickly, we are about to finish it. Will there be the splitting of anaphase 1? Of course not. There will be no splitting of centromere here. So that is the question where students, mostly they get confused. So obviously after this metaphase 1, there is anaphase 1, and in this anaphase 1, there is no splitting of centromere. There is no splitting of centromere. There is no separation of chromatid. But what will happen? Bivalent will get separated. Right? Bivalent will get separated. So you know that finally after cytokinesis, you will be having the 2 haploid cells. Because after this, whatever cells you will get 3 chromosomes there. There will be the 3 chromosomes. So yes, splitting of homologous pair will be there. You are very right, Harry Bache. Right? There will be the splitting of what? There will be the splitting of the pair. That's all. So now, what will happen in the metaphase 2? In the metaphase 2, there will be only 1 metaphasic plate, and it is the phase in which, just look at it. That's the scenario. So obviously in the anaphase 2, there will be the splitting of the centromere. There is the separation of sister chromatid. Is that clear? Is that clear? Yes or no? Is that clear? Sure? Sure? So it is going to clear all your doubts. This is the difference in meiosis. This is the difference in mitosis. This is the difference in the different metaphase stage. That's all. That's all. This is how it works. Fine? This is how it works. So just note it down, bache. Note it down. So all the points are written here. I'll share the PDF with all of you. So these are the key points that you need to take care. So just look at this particular image. Anaphase 1 of meiosis. The pair is getting separated. And in the telophase, see, initially 4 chromosomes were there. In the daughter cell, there will be 2. That's how the number will reduce to half. Okay, that's how the number will reduce to half. Rest all the points are same. Now when it comes to the interkinesis, it is just a gap. There is no DNA synthesis here. Nothing at all. Nothing at all. Even, chromosomes are not properly decondensed because they are preparing themselves for the meiosis too. That's all. This is the point here. Okay, so I hope you find it easy. So again in the prophase 2, nuclear envelope disappears. Again in the metaphase 2, chromosome will align themselves in the center. Kinetochore of sister chromatid get attached to spindle fiber. Same. Everything is same. Then in the anaphase 2, centromere split, sister chromatid separate. They will start moving to the opposite pole. And then in the telophase, nuclear membrane reappears. A cytokinesis will be there after this. Simple. And you will get 4 haploid daughter cells. That's all. This is all about the meiosis. So now we are going to practice few questions and we will quickly revise the important functions of the important organelles. So we are mainly focusing on the important points, important things from the examination point of view. Okay? Is that clear? All of you, is that clear? Done? So any doubt from this diagram? From diagram based questions used to come in the NEET exam. So you need to prepare that. Chaliye, next question. Answer this question, then we'll start the cell part. Yes, after this we'll start the cell part. So answer this question. Quickly answer this question. All of you just answer this question. Be quick. Be quick. Okay, so you can see the options. So just practice the question after this particular session. Then everything will be done. There is no need that you need to, study extra. Bas, itna sufficient hai. Okay, so here it is 1. So during cell cycle, the DNA replication will take place only once and it will take place in the S phase. Chaliye, next question. Which of the following phases are the longest and the shortest in the mitosis? Okay, tell me about the long. Overall, it is for the mitosis. Longest and the shortest phase. What about the longest and the shortest phase? Initially, you have to tell the longest, then the shortest. So, here the correct answer is this. The longest is the prophase, the shortest is the anaphase. Okay, the longest is the prophase, the shortest is the anaphase. Chaliye. So now we will be talking about the cell. So you know that cell is the basic structural and functional unit of life, because below the level of cell, there is no independent existence of life. Are you getting my point? There is no independent existence of life. When you talk about what? When you talk about the cell. Okay. So here in this chapter, which things are important? Let me tell you. All the discoveries, scientist name, function of organelles, fluid mosaic model. These are the important things. Yes, from this chapter, from this particular chapter, this is what you need to do. All the discoveries, right? Who discovered what? Yeah, even year also. So obviously, these two things are connected, discovery and the scientist name. Then function of organelles are important. And out of that, endoplasmic reticulum, Golgi apparatus, lysosome. These three are very, very, very important. Okay, these three are very, very, very important. And then the fluid mosaic hypothesis, which is given by Singer and Nicholson. Okay, it is given by Singer and Nicholson. So these are the important things. Okay, just a minute, I need to answer one question, ma'am. G2 phase for proteins, and then what about the G1? See, protein synthesis, when it comes to the protein synthesis, it is going to take place in the G1 phase also. It is going to take place in the G2 phase also. In the G1 phase, because overall cell is increasing its size, it is growing. And in the G1 phase, cell needs to form the enzymes which are even required for the synthesis of DNA. Obviously, actively protein synthesis is occurring in the G1 phase also. It will be there in the G2 phase also. Okay, it will be there in the G2 phase also. So, bache, this slide, this part is very important. Here, everything is mentioned, like Robert Hooke. You know that in 1665, he's the one who discovered, even who coined the term cell, who discovered the first cell. Okay, you know it, right? Hai na? You know it, right? See, 1665. Okay, so Robert Hooke, the English botanist, he's the one who discovered the cell. And you know that he didn't discover the living cell, he discovered the dead cell. Actually, he checked the compartments, some compartments under the microscope, right? Compartments. And for that compartments, we use the word cellulae. So we call it as cell, right? We call it as cell. So here, in the case of Robert Hooke, then when it comes to the nucleus, Robert Brown. Okay, Robert Brown in the roots of orchid plant. In the roots of orchid plant, he discovered the nucleus. Who? Robert Brown. When it comes to the cell theory, Schleiden and Schwann. Right? It is Schleiden and Schwann. What is the name of the scientist? Just write it down in the chat section. Schleiden and Shawn. When it comes to what? When it comes to the cell theory. Right? When it comes to the cell theory, it is the Schleiden and Shawn. Shawn, the sheep. Remember this? Shawn, the sheep. So Shawn is the zoologist. Right? Shawn is the zoologist. He was not the botanist. He was the zoologist. So Schleiden, botanist. Shawn, zoologist. Even it is Shawn or zoo. Remember it like this. So they gave the cell theory. I'll come to the points as well. Okay. And you know that Shawn, being the zoologist, he is the one who observed that when it comes to a cell, when it comes to a plant cell and an animal cell, plant cell is having one, extra covering other than cell membrane. So he's the one who used to figure out that. He checked it basically. He discovered it basically. Okay. And then the next discovery, the living cell, Anton von Leeuwenhoek. Another important point. Anton von Leeuwenhoek. So yes, from this chapter, from this particular chapter, you can get such kind of question. Who discovered what? Okay. Who discovered what? So this is important. Done? This is important. And the most important, how can I forget Rudolf Virchow? Hominis cellula e cellule. Hominis cellula e cellule. That is, cell comes from the pre-existing cells. Right? Cell comes from the pre-existing cell. So you guys can check it here. So when it comes to the cell, as I said, here we are talking about the unicellular and the multicellular. So you can see it here. This is the one which can live. So below the level of cell, there is no independent existence of life. Means it is the basic thing. Right? So all the living organisms, they can be unicellular, they can be multicellular, but ultimately that cell is present. You cannot say that ki mitochondria is like ki, I don't want to live with the cell, I'll live alone. No. Things like that are not going to occur. Okay, things like that are not going to occur. Clear? Is that clear? So here you can see the names as well. Anton von Leeuwenhoek. Just mark this point. He first saw the cell. He discovered a living cell. Robert Brown. All that discoverers, they are important. Okay? They are important. So, bache, when it comes to the cell theory, now let me come to the cell theory. No doubt we take the name of Schleiden and Shawn, but they didn't work together. Okay, we take the name of Schleiden and Shawn, but they didn't work together. They separately, checked the things, they observed the things, and then they put forward the point. So here, this particular part from the NCERT, it is very important. Here it is mentioned clearly. Let me mark it. See, in 1830, here the scientist name is also important. Matthias Schleiden, which is a German botanist, he examined, he checked the plants, and he observed that all the plants are having the different type of cells. Okay, all the plants are having what? They are having the different type of cells. And when it comes to the Shawn, in 1839, what he did, he checked the animal cell and he reported that there is a thin outer layer which is today known as plasma membrane. Is that clear? At that time, he was not aware that it is basically the plasma membrane. No. So at that time, he just mentioned that animal cells are covered. Animal cells are covered with a layer, and that layer is what? It is the plasma membrane. Is that clear? It is the plasma membrane. Okay. Okay. So he even checked the plant cell and he observed the presence of cell wall, which is a unique feature of the plant cell. Okay. So next question, bache. See, then Shawn, he proposed the hypothesis that the bodies of animal and plants, they are composed of cells and product of cells. So this point is very important. It has been asked as a question in the NEET examination, that who hypothesized that, right? Who hypothesized that the bodies of animals and plants, they are composed of cells and the products of cells. So this line is very important. This line is very important. Okay. No, I'm going to use the mind map, but here it is important. So together they formulated the cell theory. They mentioned that the cell is the basic unit. All the living organisms, they are made up of cells and products of cell. But they were not able to explain that how, cells arise. It was a question mark basically. It was a question mark. They were not able to explain that how that, cell arise. How they were not able to explain it at all. So later on what happened? Rudolf Virchow. This is important. This is very important. Rudolf Virchow in 1855, he explained that all the cells, they arise from the pre-existing cells. And for that, we call it as hominis cellula e cellule. Important it is. Okay. Important it is. Hominis cellula e cellule. So after this, bache, cell theory was modified. And you know what we used to call it? We used to call it modified cell theory. We even used to call it cell principle or cell doctrine. Okay, cell doctrine. Are you getting my point? So modified cell theory, also known as cell principle, also known as cell doctrine. What is it? Cell doctrine. Okay, done? Done. So you basically need rest for the next session. That's okay, that's fine. See, what are we going to do? Here in this particular chapter, only the functions are important. Mainly the functions are important. Okay, so what you have to do, you just need to read that functions again and again. You just need to revise that functions again and again. So here in this PDF, I have explained everything. I have added everything for all of you, right? We'll keep another session someday and before NEET, obviously, and we will revise these functions. Okay, with the help of mind map. So today some glitch was there. So because of that, it took our 5 to 10 minutes. So it's fine. It's okay. It's our first session. So from this session, whatever you have learned, just revise it. Just revise it as quickly as you guys can. So what is the schedule for tomorrow? Tomorrow we are going to meet at same time, right? We all are going to meet at same time at 5 and we'll be discussing the most important part of the plant kingdom as well and animal kingdom as well. And in animal kingdom, the basis of classification we are going to discuss, the tricks for that basis of classification. Along with that, the most important things from the plant kingdom, like your life cycles of algae, the examples of algae, right? Then which one is dominating, gametophyte, sporophyte. So question like that we are going to cover, okay? Fine. So I hope you all like the session. I hope you have shared this. Do subscribe our channel. You people will get the PDF in the Telegram session. You all can join it. So thank you so much. And we literally need your love, your support. And we want you people to be regular for the upcoming days so that you can get, maximum out of it. Okay. Okay. So thank you so much, everyone. Bye-bye. We'll meet tomorrow at 5. So after my session at 8, Wasim sir is going to start the session. So stay tuned for that as well. Anna, it is the bestest team ever. Isn't it? Isn't it? That's good. That's good. I'll provide the PDF. You can revise the things from this part. But tomorrow, the plant and animal kingdom. Yeah, question practice will also be there. Okay? So yes. So take care. Bye-bye. All the best. We'll share the PDF as well. So thank you so much, everyone. Thank you.